Assalamu alaikum and welcome to the Medical Concept with Dr. Falak. In today's lecture, we will explore one of the most fundamental topic in neurophysiology, that is the neurotransmitter and neuropeptide. These chemical messengers are essential for communication within the nervous system and play a critical role in everything from muscle contraction and autonomic functions to mood, memory, and behavior. Have you ever wondered why some signals in the body act within milliseconds while others produce prolonged effect lasting minutes or even longer? The answer lies in the difference between the neurotransmitter and the neuropeptide. So in this session, we will not only understand their definitions, but also explore their synthesis, mechanism of action, co-transmission, and their functional significance in both normal physiology and clinical context. So let's begin. By the end of this lecture, you should be able to define and differentiate neurotransmitter and neuropeptide. You should be able to describe the classification of the synaptic transmitter. You should be able to discuss the excitatory and inhibitory neurotransmitter and to distinguish the features of nitric oxide with other neurotransmitters. Neurotransmitter is a chemical substance that is released from a presynaptic neuron in response to an action potential which transmits signals across the synapse by binding to a specific receptor on a postsynaptic cell that may be either a gland, a muscle, or any tissue. Neurotransmitters are stored in clear vesicles. They are fast and rapid in action. The effects can be excitatory or inhibitory depending upon the type of receptor they are going to bind. While neuropeptide, they are peptide-based signaling molecules that are synthesized in neuronal cell body, released from neurons, and typically act as a neuromodulator to produce slow, prolonged effects via specific receptors. They are stored in a dense core vesicles. They can act as hormone, and they are responsible for prolonged action that lasts from days to years. Regarding classification, neurotransmitters can be classified based on their chemical nature into four main groups. The first is acetylcholine, which is unique and forms its own class. The second group, they are derived from amine and they mostly act as modulator and they include norepinephrine, epinephrine, dopamine. The third group consists of amino acid neurotransmitter and they are very fast in their action. They include gamma aminobutyric acid, glycine, and glutamate. While the last group include other unconventional neurotransmitters such as ATP, arachidonic acid, nitric oxide, and carbon monoxide. The fourth group does not follow the classical neurotransmitter mechanism. Regarding neuropeptide classification, they are classified based on their source and function and not just their structure. The first group include the hypothalamic and pituitary peptide, which mainly act as a neurohormones. They have a major role in endocrine and neural modification. They include thyrotropin-releasing hormone, beta-endorphin, prolactin, growth hormone. The second group include the peptides that act on both brain and gut, and they are responsible for linking nervous system with gastrointestinal system. They are responsible for modulation of the pain and various digestive processes. The third group include peptides from other tissues that also act as a signaling molecule in nervous system, and they include angiotensin, bradykinin, etc. Regarding life cycle of a neurotransmitter and neuropeptide, the first and the most important is the synthesis. Synthesis usually takes place in a presynaptic neuron, usually cell body or soma. From there, they are transported across the axon and then they are stored in the axon terminal. After their storage, they are released when appropriate action potential arrives, and after they release and performing their function, they are either recycled or destroyed in case of neurotransmitter or completely destroyed in case of neuropeptide. And then the synthesis, the transportation, the storage and the release, the cycle continuously takes place and the process goes on. Now for the synthesis, transport, storage and release, first talk about the neurotransmitter. The enzymes responsible for the formation of neurotransmitter, they are formed in the cytoplasm Then later they are modified by the rough endoplasmic reticulum and the Golgi body, and then by slow axonal transport facilitated by the microtubules, they reach the axon terminal. In the axon terminal, they are responsible for the conversion of precursor into their specific neurotransmitter, and these precursors are actually transported inside the cell by extracellular fluid or through blood, and this process is ATP dependent, which is coupled to glycolysis and TCA cycle. After synthesis of the neurotransmitter, they are transported inside the synaptic vesicles. This synaptic vesicles is actually driven by a proton gradient energy that is generated by hydrogen ATPase pump. These neurotransmitters are then released at synapse when stimulated by a specific and appropriate action potential, and this release is mediated by calcium ion influx. Now for the synthesis, transport, storage and release of the neuropeptide, remember that the neuropeptide precursors and the enzymes, they both are formed inside the body of the presynaptic neuron. Later on, they travel down through the microtubule tract across the axon, and then they reach the axon terminal. Here, the enzymes are going to convert or modify these pre-peptide precursors into active neuropeptide molecules, which are then again vesiculated with the same process, and upon reaching of an appropriate action potential, they are released. Now, after the neurotransmitter and neuropeptide, they have released from the presynaptic nerve ending, and they have produced their effect by binding with a specific receptor on the postsynaptic terminal or the cell. They are having three main important fates. Either they are reused or reuptake, example glutamate. Either they are enzymatically break down into their component, include acetylcholine, and either they are diffused away out of the synaptic cleft. Remember that these three fates are for the neurotransmitters, but for the neuropeptides, mostly the neuropeptides are broken down by extracellular peptidases that include neprilysin and aminopeptidases. Vesicles of the small molecule transmitters, they are continuously recycled and reused. After fusion, they release neurotransmitter, and the vesicle membrane, it becomes part of the synaptic membrane. Membrane invaginate back into the presynaptic terminal and pinches off to form a new vesicle. So new vesicle retain essential enzymes or transport proteins for synthesizing, concentrating other transmitters. Now let's summarize the major differences between neurotransmitter and the neuropeptides. Regarding size and onset of action, neurotransmitter, they are small size rapidly acting chemical substances, while neuropeptide, they are large molecule, slower in action with long-term effect. Regarding site of release, neurotransmitters, they are released from the presynaptic neuron terminal into the synaptic cleft, and same is true for neuropeptide. Regarding receptors, the neurotransmitter, they react with inotropic receptors, while neuropeptide, they react with G protein coupled metabotropic receptors. Regarding synthesis, the neurotransmitters, they are formed in axon terminal by the action of enzymes on the precursor, which later produce the actual neurotransmitter. Okay, while for the neuropeptide, the actual precursor peptide molecules, they are synthesized in the cell body, but later on they are modified into the axon terminal. So this is the main difference. Regarding function, the neurotransmitters are responsible for the opening and the closure of channels and the transmission of sensory and motor signals, while neuropeptides are responsible for a prolonged duration of opening and closure of the channels. They are responsible for the change in the number of the neuronal synapses and the receptors. They activate and can deactivate genes and affect metabolic machinery of the cells. Regarding amount release and potency, the neurotransmitters, they are released in large quantity but are less potent than neuropeptide, while neuropeptide, they are released in a small quantity but are 1,000 times more potent than small molecules. Now coming to the type of neurotransmitter. We can classify neurotransmitter depending upon the type of activation of excitatory or inhibitory postsynaptic receptors. So thus they can be excitatory neurotransmitter or they can be inhibitory neurotransmitter. Excitatory neurotransmitters are responsible for increasing the impulse transmission. They open cation channels and they cause excitatory postsynaptic potential, while inhibitory neurotransmitter, they inhibit conduction impulse, they open anion channels and they cause inhibitory postsynaptic potentials. Now let's discuss acetylcholine. Acetylcholine is a cholinergic neurotransmitter. It's released from neuromuscular junction, all preganglionic autonomic nervous system neurons, postganglionic parasympathetic fibers, and the central nervous system. The main enzymes responsible for the formation of acetylcholine is choline acetyltransferase, and it catalyzes the reaction of choline with acetyl-CoA to cause the formation of acetylcholine. There are two types of receptors, nicotinic, which are mostly excitatory, and second is the muscarinic, that can be excitatory or inhibitory. The major function of acetylcholine is in muscle contraction. It decreases the heart rate, it increases the GI motility and secretion. It's also important in memory and learning. It's terminated by the action of acetylcholine esterase. And this acetylcholine is again inhibitory and excitatory in action. Now come to the example of epinephrine and norepinephrine. So this epinephrine is released from the adrenal medulla or central nervous system in a small amount. They are actually the neurotransmitter of adrenergic nerve fibers. They are synthesized from the tyrosine. Tyrosine is converted into levodopa by means of tyrosine hydroxylase. Levodopa is then converted into dopamine by dopa decarboxylase, which is then converted into dopamine by beta hydroxylase. Then this norepinephrine is finally converted into epinephrine by the action of enzyme phenylethanolamine and methyltransferase. There are two types of receptors. Alpha receptors, they are usually excitatory, and beta receptors, they are usually excitatory or inhibitory depending upon the type of tissue. The main function is fight or flight response. They are going to increase the heart rate and blood pressure. They increases the blood glucose level. They are also responsible for bronchodilation. Regarding termination, they are either undergo degradation by deamination by monoamine oxidase, or they undergo methylation by catecholomethyltransferase. Norepinephrine doesn't have a fixed effect, right? Its action depends on the type of receptor. For example, beta-2 receptor, they cause a smooth muscle relaxation, while alpha receptor, they generally cause contraction. Thus, epinephrine and norepinephrine, they can be considered as both excitatory as well as inhibitory neurotransmitter. Then coming to dopamine. Dopamine is released from the dopaminergic nerve endings from substantia nigra, hypothalamus, and it's synthesized from the tyrosine. Tyrosine is converted to dopa, and dopa is then converted into dopamine. The major rate limiting enzyme here is tyrosine hydroxylase. Again, it has two types of receptors, D1 like receptors and D2 like receptors, right? D1 are excitatory while D2 are inhibitory. Dopamine is important for the movement control, reward and motivation, and hormonal regulation. Regarding its termination, it's either reuptaken and metabolized by the same enzymes that were used for epinephrine and norepinephrine. Regarding serotonin or hydroxytryptamine, the main source is the RAF nucleus and GIT tract. They are synthesized from the tryptophan, and the major enzyme is tryptophan hydroxylase. This is the rate limiting enzyme. Again, it has a different type of receptors. It has 5-HT1 to 5-HT7 receptors, and they are mostly G protein coupled receptors. Serotonin is important for mood regulation, for sleep, for appetite, pain modulation, nausea and vomiting, and its termination depends on its reuptake and metabolism. Then we have GABA. that is known as GABA aminobutyric acid. It's widely distributed in central nervous system. It's synthesized from the glutamate with the main action of enzyme glutamate decarboxylase, and it has two type of receptors, GABA A and GABA B. Main functions are that it's a main inhibitory neurotransmitter of the central nervous system. It decreases neuronal excitability, it's anti-anxiety, and it's important for the sedation. It's reuptaken into neuron and the glial cells, resulting in its termination. Then we have histamine. Histamine is released from the hypothalamus, and also it's produced in the periphery through mast cells. It's synthesized from the histidine with the enzyme histidine decarboxylase. It has three types of receptors, H1, H2, H3. H1 is excitatory, H2 is also excitatory, and H3 is modulatory. The main function include wakefulness, appetite regulation, learning and memory, gastric acid secretion, and it's terminated by metabolism. It's not major uptake. Now coming to the glutamate, is the most common excitatory neurotransmitter of the central nervous system. It's synthesized from the glutamine. Its main receptor include NMDA, AMPA kainate receptors, and its main function is that it's important for the learning and memory, synaptic plasticity. It's terminated by reuptake into neuron and astrocytes. Regarding glycine, it's released from the spinal cord and brainstem. It's synthesized from serine. The main receptors are glycine receptor that are chloride channels receptor, and its main function is to inhibit the motor control and spinal reflex modulation. It's terminated by the process of reuptake. Then we have nitric oxide. It's very interesting and it belongs to class 4 neurotransmitter. It's released from the endothelial cells and the neurons by the arginine that is responsible for its production, and the main enzyme is nitric oxide synthase. There are no classical receptor for nitric oxide, but it mainly acts via cyclic GMP method. It's responsible for the vasodilatation, neurotransmission, and memory. It's terminated by rapid diffusion and breaking down. This is another important concept, that what is the main difference between nitric oxide and other neurotransmitter. Other neurotransmitters are formed as preformed, or they are stored in vesicle in a presynaptic neurons, while nitric oxide is synthesized instantly as needed and it's not stored as vesicle. Other neurotransmitters are released as vesicular packet only from the presynaptic neuron, while nitric oxide it diffuses from both presynaptic or postsynaptic neuron. It's also considered as a retrograde neurotransmitter. Regarding other neurotransmitters, they enter postsynaptic neuron by attaching to the receptors or the ion channels, while nitric oxide diffuses into the presynaptic or even postsynaptic neurons for its action. Other neurotransmitters have a specific function that were discussed previously, while nitric oxide, it activates guanylyl cyclase and it causes changes intracellular metabolic functions by the modification of neuronal excitability for seconds, minutes, or even more. We have another very important concept. This is known as co-release and co-transmission. First, what is co-release? When two or more neurotransmitters, they are co-localized within the same synaptic vesicles and they are released together upon stimulation. This process is known as co-release. Examples: serotonin and glutamate, acetylcholine and ATP. We have so many examples for co-release. Second concept is co-transmission. Co-transmission is that when one neuron uses more than one neurotransmitter to communicate. In this case, neurotransmitters are stored in different vesicles but within the same neurons, and then they are released under different conditions, right? Release may vary depending upon the calcium levels or the firing frequency or the frequency of action potential, right? So co-transmission, it enables flexible and activity dependent modulation of synaptic signaling. We have an example of substance P as a neuropeptide. Neuropeptide main role is in pain transmission. It's primarily released from the primary afferent neurons, especially in the spinal cord. It's also released from the central nervous system and from other peripheral tissue. Its main function is in pain signaling, vasodilatation, mood and stress modulation, and nausea and vomiting. So in short, we can say that excitatory neurotransmitters include glutamate, aspartate, acetylcholine, norepinephrine. Inhibitory include GABA glycine, while both we can add acetylcholine, dopamine, serotonin, norepinephrine, histamine, right? There is another important concept of the neuromodulator. By definition, signaling molecules that modify synaptic transmission and neuronal excitability, often via slower and longer lasting mechanism. They can affect multiple neurons simultaneously. They may act as a neurotransmitter, neuropeptide, or other signaling molecules. The main function of neuromodulator is that they regulate the synthesis, the breakdown or reuptake of the neurotransmitter. They excite or inhibit postsynaptic receptor either independently or in conjunction with other neurotransmitter. They modulate gene expression and also regulate local blood flow. There are three types of neuromodulator. Number one is the monoamines. They include dopamine, serotonin, norepinephrine. The second group include neuropeptide that include opioid and non-opioids, while other group include nitric oxide and endocannabinoids. So in summary, neurotransmitters and neuropeptides is not just about the chemical release, it's a highly organized and dynamic system. We have seen that neurotransmitter differs in their synthesis, storage, receptors and mechanism of termination. Small molecule transmitters, they act rapidly and are often recycled, while neuropeptides, they are synthesized in the cell body and they produce slower prolonged effect. We also explored important concepts like co-release of co-transmission, which highlights how a single neuron can fine-tune its signaling depending upon the physiological condition. Finally, remember this key principle, right? That the effect of neurotransmitter is determined not only by the molecule itself, but by the receptor it activates. So understanding these concepts provide the foundation for interpreting normal nervous system functions as well as many clinical condition and pharmacological intervention. So thank you for watching. Inshallah, we will meet next time again. Thank you.